Today, Tracy Johnson, a research entomologist with IPIF, will be presenting his research on biocontrol of weeds in Laupahoehoe forest. His research is being done through a permit with the HETF. Please note that permits are required when entering the HETF for research purposes. And whenever you're ready, Tracy, you can start.
Okay, thank you, Olivia.
Thank you.
Yeah, I'll share my screen. All right. Yeah, so yeah, I'm an entomologist. My job is to do biological control of weeds research, specifically trying to tackle invasive plants in Hawaii and Pacific Islands affiliated with the US. And a lot of our big problems and the targets that I work on actually are present and reflected in Laupāhoehoe forest, unfortunately. So that is one of the places I hope to serve over the years. We started monitoring guava there almost 10 years ago to get a clearer idea of how the invasion is proceeding. And so we have some study plots set up across some different elevations, from down towards the edge of the forest, a little bit above the edge at 2,500 feet, where the guava is really thick and there's lots of clidemia and ginger as well. So a lot of invasives at that level. And then as you go up, the forest gets more and more native. So part of the reason was to understand the invasion across that gradient of heavily invaded to lightly invaded, and then to prepare for when biocontrol becomes available and see how the impact of the biocontrol plays out when you have heavy infestation of guava, for example, and then lighter infestation. So we have been monitoring those plots. The initial round of measurements was in 2013. And so as you go up in elevation, there's a lot less guava, so it shows in this graph in a couple of different ways. The basal area is just measuring the thickness of the stems, and there's a lot of stems down low, as most of you know if you're familiar with the forest. And then you go up and it thins out and there's a lot less, fewer stems and smaller amounts of guava. Unfortunately, it's increasing. It's increasing pretty fast. When we remeasured these same plots in 2016, there's a lot more guava stems and a lot bigger guava stems. They're growing fast. So guava, and it's pretty much on par with other places where we measured guava, basically increasing at 10% per year, which is fast for a plant. So guava is invasive. There's not much news there. It's growing fast. It's also, it hasn't gotten as bad, or I should say, it's going to get much worse if we just leave it. This higher elevation it's only about 3,000 feet, and more if you look on the Puna side of the island. Guava is well past, it's really thick guava, well past 3,000 feet now. So it's just marching up the mountain. So part of our objective with biocontrol is to try to find some management that can slow it down. So I'll talk a little bit about what biocontrol is and how we do it. And our group is involved in all the different phases of biocontrol. So it starts with picking a target. And we have to prioritize because there are a lot of invasive plants, but not all of them are good targets for biocontrol. And then we work in the country where that plant comes from to find natural enemies that look like they're specific. So there's a lot of research that goes on abroad in the countries where the weeds come from, looking for natural enemies that evolved with those weeds. And then if we find something that we think is good, that we've tested in a foreign country and looks like it's promising, then we'll bring it to Hawaii and test it in a quarantine lab, which is a secure facility where the insects can't escape, but we can test them with Hawaiian plants, make sure that they're very specific. And then finally, if all the data looks good, and it's a natural enemy that we want to release, then we propose that through the state and federal regulatory agencies, and it gets very thoroughly reviewed. And then if it passes, then it's released and we monitor for the impacts. So there's a lot of steps involved. The foreign work is challenging. It's very interesting too. A lot of collaboration necessary to make this happen. And so I'll talk more specifically about some certain projects, and I'll start with the strawberry guava. So strawberry guava, we did find an insect in Brazil. The guava's from Brazil, and the insect did pass all its reviews, and we released it in the state in 2012. It's a tiny insect, so when we initially release it, we're just using these little paintbrushes and putting it very carefully on the plants. It's a gall-forming insect, so it starts out making galls and they grow with the leaves. So it has to have the new leaves to make galls. The insect, so I'll talk a little bit about the biology. Each of these are two galls. The fatter one is a female gall. The female's inside there, and she's stuck in that gall for her entire life, reproducing in there. So she's immobile, basically. She'll generate eggs, and the eggs can be pushed out of the gall, or they can hatch and crawl out. The eggs can also be embedded in a waxy substance that she secretes, and that can be blown. So the main way that this thing can disperse is by wind. They can also crawl. So once the eggs hatch, they crawl, but they're tiny, tiny things. So they don't crawl very far. But they seek out the new leaves and settle on those new leaves. And as those leaves grow, the gall forms around them. So you can see in this close-up, the leaf tissue starting to form around the insect. So that's an interesting thing about these gall-forming insects, is that the gall, their home, is plant tissue. And so they are causing the plant to make a structure that they can live in and feed in. So it's a very close relationship where they're able to communicate with the plant and force the plant to do what they want. So it's a relationship that doesn't shift very much in evolutionary time. It takes a long time for a very intimate relationship like this to evolve. So it's a very highly specific sort of insect. So this is one of the release sites that we put this insect out initially in 2012, was a little orchard of guava trees. Sorry, I went too far. And we measured these trees very intensively. And just to summarize briefly, as the trees become more and more galled over time, and they're exposed to galling for a longer period, we're starting to see their growth slow down. So in these graphs, the on the left side of the graphs is not very much time with galling, and then a few years as you move to the right. So with there are 3 different varieties of strawberry guava, it's important to note, and we're studying all of those to make sure we understand how this insect interacts with all of them over time. And we're seeing slower growth for the red and the yellow. Not so much for the spindle. That's a big tree. It takes longer for the impact to unfold. On the Big Island, the main invader up into the forests, the higher elevation forests, I should say, is the yellow variety. So the impact looks good on that. It actually, the impact seems to unfold fastest on the yellow. There is some red variety too in Laupahoehoe, but it's mostly that yellow one. And as the trees start to grow slower, eventually we're starting now to see lower fruiting too. So, and this is the impact we want to see. We want to limit the spread of seeds from the invasive tree. We don't see this insect killing trees. That's not what happens really in Brazil. But by producing all these galls, it does divert energy away from growth and fruiting. So we're really hoping to suppress the trees over time. And it does look like that's happening with the red and yellow varieties. The spindle is going to take longer. It's pretty clear that's a robust tree, and I don't have any reason to think that it won't eventually have some impact, but it certainly is taking longer than 8 years to see an impact on that tree. And for all of them, this is a gradual impact. 8 years on these trees that we've had it the longest. We've also been working to develop ways to spread this insect around. As I said, it's not very mobile. Doesn't fly to the new trees. It can get blown, but it doesn't really control where it gets blown. So to get it around all the places in the state, we needed good ways to establish it. And one of these is shown on the left, getting stems of the heavily galled and putting them next to a new tree. Often we'll cut the trees and get new sprouting so that there's lots of new leaf tissue for the insect to move on to. Recall it needs to have that new growth. A leaf that's already fully grown, the insect can't do anything with that. It has to have the brand new leaf to start to make galls. But what we found is we can also just spread it by taking an individual leaf, and this is one technique we developed, is tying it with some guava stems for weights and throw it up into a tree, get it up into the canopy more quickly. So we know that works. And we're moving now into trying to develop aerial methods for spreading it, because there's a lot of guava in the state. It has been released in lots of sites. This shows some of them, some of the initial forest release sites around the state. It's establishing everywhere we put it. It's just now getting to Laupahoehoe. It hasn't been there until now, I think. But yeah, to get it everywhere, we're going to have to be more active to place it. We have done some recent work, very recent work, to try to figure out how far it can spread on its own. And so measuring from where we initially put it in Waiakea, as I said, now 9 years ago, it's appears that it's spread 2 kilometers, which is about a little over 200 meters per year, which is farther than I thought initially. We can't be totally sure that there weren't some other sites in some of this area where it could have spread from, but that's a believable rate of spread with a good open environment. In a more closed forest environment, I'm kind of expecting it's not going to spread as fast. So to get it deep into a forest, I think we're going to have to put it there more actively than we have been up to now. So we're working on trying to get that done through aerial means, and that's looking at the scale of the whole eastern Big Island. There's a lot of forest, and a lot of forest invaded by strawberry guava. And the other thing we're hoping to do in our Laupahoehoe, as we get the biocontrol going in there, we want to understand how to integrate other restoration activities. So like, it's great, you can potentially slow down the guava, prevent it from getting worse, but can you restore an area that's already invaded with guava? So we're going to explore some ways to kill off the guava once we get the biocontrol growing, and see whether the biocontrol can protect a restoration plot from more invasion of the biocontrol. And I do expect that it can work that way. It's going to be gradual and take time, but hopefully we can see that happen over the next several years as we work in Laupāhoehoe and other sites. Another weed I'm going to talk about more quickly is Himalayan ginger. We've seen this also increasing in Laupāhoehoe. That's this picture is from the national park, but it grows in these very thick stands over time, pretty much shading out all the native understory. So it can come to dominate in a fairly short amount of time our wet forest understories. So we've been collaborating with folks from Great Britain to work with people in the native range mainly most recently in India. And so this is the plant in its native range in the Himalayan region. And you just find scattered stems with lots of other vegetation. And there are certainly our natural enemies feeding on it there. And the natural enemy that we've decided to prioritize is this fly that it lays its eggs in the sheath of the stem as it's developing. And the larvae bore into that stem. And the photo on the right shows I think it's a fully mature after the larva has pupated inside the stem. So, but it'll feed on the very center of the growth of the stem, potentially and often killing the apical growth. So that means that you have a stunted stem. It doesn't get to reproduce, which would be perfect for controlling our invasions. And the reason this fly has been prioritized, it looks like that there's a species of this fly that's very specific to the species, the Himalayan ginger that we have here in Hawaii. So the tests are still happening with this insect. We haven't gotten it here in Hawaii yet to test in our laboratory. And we're collaborating with folks in New Zealand as well. They have been supporting the work by the researchers in the Great Britain. And they're probably going to be working with this fly before we do, and then we'll get flies from them. So that's the plan. Unfortunately, I haven't been able to get any flies in the COVID year, and so things are slowed down a lot in the biocontrol world in terms of international collaboration for obvious reasons. And then the last weed species I want to really focus in on is clidemia, also known as Coster's curse. I've got some right in over my head here. It is a weed that the state has worked on biocontrol for a long time. The first insects were released in the 50s, thrips, which had good impact. They really reduced densities of the weed in open, sunny environments like a pasture. They tend to be pretty effective. And then more recently, some other natural enemies have been released. There's a fungus that attacks the leaves, which does seem like it's effective, but only in very patchy, limited situations. So we do see outbreaks of it every now and then, but it doesn't seem to wipe out populations of the weed. There's also a leaf mining beetle that's been released, and a moth, a tiny moth that bores, its caterpillars bore into the fruit. And we've been monitoring these more recently to get more information. The leaf miner, unfortunately, seems to have a lot of natural enemies that has natural enemies that really impact it in Hawaii. So its populations seem to always just stay low. The Mompha I'll talk a little bit more about in a minute. But the take home for us is that we need other natural enemies. And I've been working for years now on trying to develop other natural enemies. And one of them that looks promising is this gall wasp that attacks the fruit of clidemia. It's a little tiny wasp. It lays its egg in the flower, and then the larvae develop inside the fruit, kind of taking over the normal development of the fruit. So instead of a soft purple fruit, you get this enlarged green hard fruit, and inside it's instead of getting normal seeds, you get these big galls that are containing the wasp larvae. So that is we've done some testing of that. We're not finished with testing yet, but it's almost certainly very, very highly specific. There's actually another wasp related that attacks Myconia in the same way, and we're working on that one too. Another natural enemy that I think is really promising is a nematode that galls any new growth, including like flowers and developing fruit. So these growths are galls, and inside these little growths, you have little microscopic nematodes developing. So this one's been challenging. We don't currently have any facilities for this kind of work here in Hawaii. So we're having to do it all outside of the state. And so that one remains under development as well. So, but I have, I think it's encouraging. It really thrives in wet environments. So exactly the kind of wet forest understory that where our clidemia is really out of control right now. And so I'll just go a little bit into one of our recent studies on Mompha, the caterpillar. It's a moth that lays its eggs, tiny eggs on the outside of the flowers and fruit, and then they hatch and they go into the fruit and they develop from small caterpillars to big caterpillars. The final stage, they have these red stripes, and they come out of the fruit and then pupate either on the plant or in the ground. And pretty much they're present in all stages of from the small tiny flower buds to the green fruit. By the time the fruit get mature, they'll have come out. So hopefully they are slowing the growth and possibly limiting the numbers of fruit developing. We don't know that for sure yet. But certainly eating some seeds. We don't think they're eating enough seeds to have an impact though. If you ever have taken a ripe clidemia fruit and squashed it, there are about 600 or 700 seeds in there. And if this insect is only taking out 50 or 75% of those seeds, we don't expect that really to be enough. We need something that would be wiping out 90% or more of seeds. So that's probably why, one of the reasons why we still have a lot of clidemia. It still is kind of early days since 1995 when this was released. But we've looked around and you can find this insect pretty much everywhere statewide now where there's clidemia. We looked across elevation to see if there is any effect of elevation, and it does seem like it's pretty clear that the insect is less abundant at higher elevations. So this 980 meters, that's about a little over 3,000 feet on Laupahoehoe. So up there, there's still not very much clidemia yet, but it's tending to escape the impacts of this insect by being up at that elevation. Why that's the case, we don't really know. It doesn't totally escape it though. In certain times of the year, we are finding it. So, I mean, we can hope that impacts from this insect get bigger and better over time, but the fact that it is widespread and we're still getting a lot of reproduction of clidemia still suggests that we need more natural enemies. And then just real quick, yeah, since I've got some time, I'll talk about some other targets. Tipicina, we have that growing up into Laupāhoehoe as well. We have a flea beetle that's really specific to mellostomes. So this kind of gives you an idea of what kind of testing we do when we look at these. If you look at all these different species of plants, especially on the other mellostome species that we look at, and then other related plants and then completely unrelated plants, the substantial amounts of feeding only occur on a few species that are in the same family with Tipicina. And that family is mellostomes, like clidemia, myconia. All mellostomes in Hawaii are non-native, and pretty much they're all weeds. So so this has a good level of specificity considering what we want to control in Hawaii. So this insect is already in the process of getting approval for release. Hopefully that process is going to move forward this year, and if we can get back to Brazil, get insects from Brazil sometime next year, then we can think about releasing this insect. And then Himalayan raspberry, unfortunately, that's spreading on this island a lot and definitely has footholds in Laupahoehoe. We've been working with collaborators in India and China to look at natural enemies there. There's some fungi, some insects. This one is really, we have to be careful, obviously, with this one because we have native Akala in the same genus. So we really have to find something that's very, very specific to contemplate biocontrol of this. But we're looking. And then Albizia, that's another big project that we're working on. There are natural enemies in Indonesia and Papua New Guinea. We've prioritized a few of those and begun working with collaborators there to study these species. We're still a ways off from working with them here. Faya, also unfortunately increasing on the Hamakua coast. This is what it looked like some years ago in the national park. There was a natural enemy released in the 90s for Faya biocontrol, but it's this little moth. It has suffered a lot of impact from natural enemies, so it's not at all effective. And so we're looking at other natural enemies. One possible insect is this weevil from the Canary Islands, but no studies of this started yet, so still early days. There are fungi on Faya in its native range too, so potential for the future, but there's still a long way to go in terms of the research. So that's it. That's where I think I'll stop, and I think we have lots of time for questions. So Olivia.
Thank you so much, Tracy. There's no questions in the chat, but if anybody has, there's one in the chat, but feel free to put them in the chat, or after I ask this one, feel free to unmute yourself and ask that way. But let's go. Chuck Premier asks, in previous discussions with Pat Billy, Wiley, okay. I recall him mentioning that what we refer to as Hedychium, I'm butchering that.
Hedychium gardnerianum. Yeah.
Thank you. Maybe hybrid origin and possibly not true.
I don't know if we've gotten final confirmation on the status. I've heard that as well, that there's some hybrid, so that would mean that the particular species that we have here is not actually pure Hedychium gardnerianum from wherever, but it's been bred with other things and it's a hybrid. They are testing our ginger. They have been testing our ginger in the UK. So hopefully any insects that they develop will be effective against ours. But yeah, that is a concern. You want to know, ideally you want to know as much as you can about the invasive plant, that its genetics, to target where specifically it came from, that you're looking on the same critter or same plant when you're in the native range. But yeah, that does complicate things for sure. I'm sorry, did I address his question?
My bad. I think so.
Hi, Tracy.
Yeah.
This is Chuck. Yeah, you answered it perfectly. Thank you.
Okay.
Awesome. And Susan says, great presentation. Are you still monitoring the Laupahoehoe plots? Your data point showed today. Yeah.
Yeah. So we're overdue to get back in there and measure again. So that's my intention this year. It's a lot of work to hike up there and measure those things. But yeah, so since the biocontrol has not been operating, I'm just expecting, I'm pretty confident we'll see that the strawberry guava has increased. Hopefully this year we can get the biocontrol started enough so that it starts having impact, but based on what we've seen elsewhere, it takes time for the populations to biocontrol to build up, and then more time for that high level population to begin to have impact. So we're talking about 8-10 years for when you start really seeing the impact. So it's a very gradual process. So we're gonna need some young folks. After like Susan and I have retired, we need some more people to carry the stuff on, because it's gonna take a while to see all of this unfold and really tie it in thoroughly with a comprehensive restoration approach, which is really necessary.
Awesome. Kyle Kagimoto says, thanks for the updates, Tracy. Any way that us field techs can help out? And also any talk of doing biocontrols for blackberry on Kauai?
Blackberry on Kauai? Yeah, I'm not sure about the status of that. I'm assuming you're talking about our Rubus argutus. But yeah, if there are biocontrols that were released in the past for that. Unfortunately, that was back in the 1960s, and those were biocontrols that weren't as specific as we would like them to be today, because back then they didn't mind if a akala got chewed up a little bit. So the biocontrols that are currently in the state will eat akala. They don't seem to have a huge impact on it, but we do have some biocontrols that you can find on akala. There are also some invasive insects that also eat akala. But yeah, I'm not sure what the status is on Kauai. Sorry about blackberry. Help? Yes, you can help. So I guess we could use some help with the monitoring. If anybody has time and inclination to help us measure some guava in up Spencer Trail in the next few months, yeah, I could use some help.
Awesome. Sorry, there's construction going outside of my window, so sorry if anybody can hear that. There's another question by Eileen Yee asking about biocontrol on fiddlewood.
Okay, yeah, heard that before. Fiddlewood's increasing. There's a lot of fiddlewood, and we get calls for that from Oahu too. I have not worked on fiddlewood at all, and it's really hard to see me getting very deep into that before I retire in 7 or 8 years. So yeah, this is the other thing we kind of need. We need more people doing research for biocontrol of invasive plants in Hawaii, invasive arthropods as well. Our our infrastructure is limited in terms of our facilities. We're trying to develop new facilities, and our staffing basically has been cut in half over the last 20 years. And most of the staffing used to be with the state, and just through attrition and budget cuts and flat budgets, there's basically half as many people or half as much research effort, like permanent people working in biocontrol as there used to be. Whereas we need to be going in the opposite direction. We have so many invasive species problems. We need to do more. We have to have more capacity for biocontrol. So yeah, it's hopefully we're starting to develop some momentum for turning that around. I think our state biosecurity plan and state priorities are hoping to shift that in the right direction.
Awesome. There was another question by Jamie Miller about biocontrol agent Aroplecturus dermis for the stinging nettle caterpillar. Wondering if there's any follow-up.
Yeah, good question. I don't know the answer to that one. We could call up Pat Conant. He's retired too, so don't bother him actually, but I don't know if the state has done any follow-up, but that one looked like it was working very well. But I have not been working on that one. That was a state project. And so if you still have problems with stinging nettle caterpillar, you can let me know, or I can point you to the right people at the Hawaii Department of Ag to talk to. But I believe that was a very successful, that it was a parasite that attacked the caterpillars. I believe it's very successful. So yeah, does anybody else have a experience or update on that? I don't know. There's a lot. So let me just talk quickly to the students out there. Like our project with MONFA, the follow-up we've been doing with MONFA, I mean, the state doesn't have the staffing to follow up and do like detailed studies. So if you're interested in entomology or plant pathology or ecology of natural enemies on plants, there's a lot of master's degrees waiting to be done on systems that are already in place in Hawaii. We just haven't monitored to know really what's going on with them because there's not enough staff to do that. So maybe the stinging nettle caterpillar is an example, but there are a lot of past cases where we could learn more from what has already been done.
Awesome. That also kind of brings me into this other person's questions, Bill Hester. If people have more questions, can I provide them with your contact information in the follow-up email, Tracy?
Yeah, certainly. I meant to put my email down, but
no worries.
Yeah,
I can totally put it in the follow-up email. But we still have time for a couple of more questions. Susan had to leave, but she did put, is there any biocontrol agents for banana polka? It has been getting pretty bad in Puvawa.
Guava. Oh yeah. So I haven't worked on it since I came here. That we had some agents kind of in the pipeline 20 years ago, but we didn't really have the staff to keep that project going. There were some biocontrols released in the 80s and 90s, and the main impact is from the fungus that appear to be really successful in Laupahoehoe, really knocked down in that those upper elevation infestations of banana polka, really knocking that back. A very highly specific fungus. The story that I've heard is that might not work as well, and it makes sense it wouldn't work as well in a drier environment. So I believe there's places on Maui and shoot, I'm blanking. Kualalai, the slope like Puvawa, where it still can be a problem. I don't have a solution for that yet in terms of biocontrol. So yeah, that may be an ongoing challenge. But there's also the other possibility is that it just takes time, and eventually there can be an outbreak of the fungus, because a lot of these biocontrols work kind of cyclically. So they may be in the environment, and the weed may get away from them temporarily, but then they find it, and if the conditions are right, there could be an outbreak. The conditions for the fungus have to be right. So if we have I guess pray for a wet season, maybe in Guava, maybe the fungus could take off. I don't know.
Awesome. Yeah, does anybody else have any questions or comments? Mark Wasser says in the about Polka has also gotten bad at Hawaii Volcanoes National Park, and especially in some mesic areas post-fire on the Mauna Loa Strip. But
yeah, that's interesting. Yeah, I mean, these weeds they're with us for the long haul. And so the biocontrol it's the long haul solution, but it may take time. That we could contemplate like reintroducing, I guess. That would be possible. I don't have a lot of experience with the Septoria fungus on banana polka, but I do know that Steve Bergfeld worked with Trujillo in the early parts of that. And one of the ways they spread it was very simple. They made a lei with infected leaf material and put that out in a new site. So I don't know if that sort of proactive approach would be helpful. It could be contemplated.
awesome. I can give a few minutes if anybody else would like to ask any questions.
Hi, aloha. My name is Desmond. Question is for Tracy. Mahalo for your presentation. Thank you very much. And mahalo for the work you do for the info and stuff. But I just want to know, you know, from the beginning of time when biocontrols were started to get introduced till now, and you know, from that time till now, and how much biocontrol has been introduced, what is the success rate? What would be probably the percentage of success rate that even using biocontrol has had?
Mm-hmm. Yeah, that's a great question. So the beginning of time is basically early 1900s starting people doing weed biocontrol. So we've got about 100 plus years of history. And some of the analyses I've seen, people will rate as a high as like 30% success rate. It's definitely not a high. It's not above 50. So, and it could be well below 30%, depending on what kind of numbers you're looking at. So that's a good point to remember, that even though we introduce something that's very specific, a lot of the time something might go wrong with the way it impacts the plant. So there could be interference from natural enemies of its own, for example. That definitely occurs in Hawaii with caterpillars. So we can't guarantee that we're eventually going to have a solution from biocontrol for, say, Myconia. We're working hard to try to come up with good options, but it's possible that not all the time, it's not always going to work out for us. So biosecurity is important. We don't need any more problems. Yeah. Yeah. I mean, that's it. I hope we don't end on this downer note, but it just doesn't always work out super successfully, at least historically speaking. I guess on a bright note, I guess sometimes it takes quite a while to find the right natural enemy to have the impacts that you want. And so that's why often these projects last 10, 20 years more or more. And the more you look and the harder you look in the natural, the native range of the weeds, the more different natural enemies you discover. So that's another reason to support preserving rainforests in Costa Rica and Brazil, because they've got natural enemies there for our weeds. And we need that biodiversity.
Right on, Tracy. Mahalo. Thank you very much.
Mm-hmm.
Another question, I think this might go for anything. Is there any biocontrol that you handle? Is there any legal or ethical restrictions on moving organisms or biocontrols and infected materials around, specifically like the fungus that you were talking about?
Yeah, I'm not sure which fungus, but definitely there's legal stuff for sure. There's international legal stuff, and to a certain extent it gets into ethical an ethical realm too. For example,
hey Tracy.
Yeah.
That was my question, Smart Wasser. I wanted to caveat that with for biocontrols that are already present on the landscape in Hawaii, like say moving Tectococcus around or something like that.
Yeah. Oh, so moving Tectococcus, I mean, Tectococcus is approved by the state for, I mean, its introduction is approved. So that's basically the state regulates the initial introduction of it, and then basically it's available for management. So it's kind of up to managers whether they want to utilize it or develop it. I mean, it will spread over time, but we're talking about many decades to get into deep forest areas of if we're just going to rely on it spreading on its own. So the guava is going to get way worse unless we get more active about dispersing it. Yeah, so I guess without being a lawyer, I think it's okay to move Tectococcus around. I've been working to show people methods for doing that so that they do it effectively. We are moving it in galled foliage, right? So we have to be cognizant that of anything else that's on that foliage, like specifically fire ants. Please don't move LFA around. But basically, we're not finding any parasites so far. I'm talking about Tectococcus, but there are natural enemies that are generalists. So like lacewings and lady beetles will feed on the crawlers. So you'll find those things, and they're kind of everywhere anyway. But it behooves us to be careful when we're moving things around, that we're only moving around what we intend. So if anyone needs like hands-on help getting better at that, I'm happy to provide some information or meet up with you.
Awesome. Thank you so much, Tracy. It's 1 o'clock, so we can start signing off now. Thank you everyone for coming.